Hello everyone. Welcome back to our Plante. In today's lecture, we'll learn all about the life cycle of angiosperms, part 4, in a simple and easy to understand way. So, let's begin. The life cycle of angiosperms, part 4, development of female gametophyte. The nucleus of the megaspore divides into two, and the nuclei are separated from one another to the opposite poles by the formation of a large vacuole between them. Each daughter nucleus divides twice, resulting in the formation of 4 nuclei at each pole. One nucleus from each polar group migrate to the center. These are called polar nuclei. They unite to form fusion nucleus, or secondary nucleus, which is diploid. The 3 nuclei left at the micropylar end constitute the egg apparatus. One of them, which is away from micropyle, is the ovum or oosphere, while the other 2 are the synergids or help cells. The help cells are considered to help in the process of fertilization. The remaining 3 at the chalazial end of the embryo sac are organized into small, naked or thin-walled cells and are known as antipodal cells. They usually degenerate, but in some cases they are active and divide to form a tissue. They are supposed to be nutritive in function and supply food till the formation of the endosperm. Development of female gametophyte, types of embryo sacs. The above-mentioned development is that of a normal type 8-nucleate monosporic embryo sac. However, there are several variations from this normal type, and the embryo sacs are divided into categories on the basis of these variations. A monosporic type. The embryo sac of this type may be 4-nucleate or 8-nucleate. 1. Polygonum or normal type, already described under the heading of development of female gametophyte. 2. Oenothera type. It is characteristics of the family Agavaceae. The megaspore nucleus divides twice and produces 4 nuclei at the micropylar end. 3 of them act as egg apparatus, while the 4th one is the polar nucleus. The second polar nucleus and the antipodal nuclei are absent. Bisporic type. The bisporic embryo sac or 2-nucleate dyad degenerates after the first division. One of the dyad degenerates. 1. Allium type. In this type, after the first division, the mature sac has 8 nuclei. The other dyad forms 8 nuclei by successive divisions in Polygonum type. This type is usually found in Liliaceae, Amaryllidaceae, and Orchidaceae, but is less common in dicots. Endymion type. After the formation of dyad, the lower cell may disintegrate. Its nucleus divides once or twice, producing 4 nuclei. C. Tetrasporic type. 8 tetrasporic types are known. In these types, protoplasts of all 4 megaspores build together the embryo sac. The number of nuclei in the embryo sac varies from 4 to 16. Peperonia type. Each of the 4 megaspores' nuclei divides twice, forming nuclei which are uniformly distributed along the periphery of the embryo sac. 2 nuclei at the micropylar end form an egg and a synergid. 8 of them fuse to form secondary nucleus, while the remaining 6 remain at the periphery of the embryo sac. 2. Pinea type. In this type, the 16 nuclei are arranged in quarters, 1 at each end of the embryo sac and 2 at the sides. 3 nuclei of each quarter become cells, while the 4th one moves towards the center and act as polar nucleus. Therefore, there are 4 triads and 4 polar nuclei. 1 cell of the micropylar triad is the egg, and is the only functional cell. 3. Drusa type. The nucleus of the megaspore mother cell divides meiotically, producing 4 nuclei. 1 daughter nucleus moves towards the micropylar end, while the other 3 move towards chalazial end. Each nucleus divides twice to form 4 nuclei at micropylar end and 12 nuclei at the chalazial end. The 4 micropylar nuclei give rise to egg apparatus and upper polar nucleus. and the 12 callosal nuclei give rise to 1 lower polar nucleus and 11 antipodal cells. Chrysanthemum cinerariifolium type. The 4 megaspore nuclei show 1 plus 2 plus 1 arrangement, i.e., 1 nucleus lie at each pole and the 2 in the center. Megaspore nucleus at micropilar end divides twice, forming 4 nuclei. The nucleus present at the callosal end shows no regular division. The embryo sac may have 6, 9, or 10 nuclei. Fritillaria type, 8 nucleate. 3 out of the 4 megaspore nuclei move towards the callosal end, and the 4th one towards the micropilar end. The 3 nuclei present at the callosal end fuse to form a triploid, 3N nucleus. The 3 nuclei at micropilar end, and the triploid, 3N nucleus at the callosal end. At the end of the division, there are 2 nuclei at micropilar end and 2 triploid nuclei at callosal end. The nuclei divides once, migration to the center, forming a 4N secondary nucleus, while the nuclei at the micropilar end form the egg apparatus, and the remaining 3 nuclei at the callosal end form antipodal cells. 6. Plumbagella type, 8 nucleate. The 4 megaspore nuclei show arrangement, i.e., 1 nucleus at the micropilar end and the 3 nuclei at the callosal end become binucleate. Both the nuclei at micropilar end and the 3 nuclei at the callosal end fuse to form triploid, 3N nucleus. In this way, the triploid, 3N nucleus at the callosal end, 1 nucleus from each end migrate. Plumbago type. The 4 megaspore nuclei arrange themselves around the embryo sac. 1 at the micropilar end, 1 at the callosa, 1 at the right side, and the 1 at the left side. The nuclei divide and 8 nuclei are formed, arranged in 4 pairs. 1 nucleus from each pair migrate to the center and fuse to form tetraploid, 4N, secondary nucleus at the micropilar end form the egg, while the remaining 3 nuclei, Adosa type, 8 nucleate, 4 megaspore nuclei divides to form 8 nuclei. 3 arrange themselves at the micropilar end and form the egg apparatus. The other 3 are arranged at the callosal end and form the antipodal cells, while the remaining 2 are the polar nuclei. Fertilization. The pollen tube, after entering the embryo sac, bursts, and during this process, one of the synergids is also destroyed. The tube nucleus disintegrates. One of the male gametes moves towards the egg and fuses with it, syngamy, and the other male gamete fuses with the secondary nucleus, triple fusion. This process is known as double fertilization. Double fertilization is the characteristics of angiosperms. The second synergid is destroyed after fertilization. The antipodal cells also disintegrate, either before or after fertilization. The oospore develops into embryo, while the endospermic nucleus develops into the endosperm. Development of embryo, embryogenesis. The fertilized oosphere secretes a thick wall around it, and is transformed into an oospore, which acts as the first cell of the sporophytic generation. The oospore, after undergoing a period of rest, few hours to few months, develops into an embryo. Generally, division of nucleus is followed by wall formation. Early stages of development of monocot and dicot embryos are the same, but later stages show a marked difference. Development of dicot embryo, Capsella bursipastoris. The oospore elongates and divides transversely into a suspensor cell towards micropilar end and an embryo cell directed towards the cavity of the embryo sac. The embryo cell enlarges and becomes spherical in outline. It divides by a vertical division into 4 cells. 2 further division, one vertical and the other at right angles to the first one, result in the formation of an octant, 8-celled embryo. This is the hypophysis, or posterior octants, next to the epibasal, or anterior octants, towards the callosal end. The 4 octants towards the chalazal end of the embryo is called proembryo. The epibasal octants give rise to plumule and the cotyledons, while the hypobasal octants form the hypocotyl root end of the embryo, except for its tip. Now all the 8 cells of the octant divide periclinally, forming an outer and inner layers of cells. The cells of outer layer divide anticlinally, forming a peripheral layer, the dermatogen, from which epidermis is formed. The cells of inner layer divide longitudinally and transversely, forming the periblem, ground tissue, beneath the dermatogen, and plerome, vascular tissue, in the center. The dermatogen is incomplete at the root end of the embryo because the apex of the root is formed from hypophysis of the suspensor. The hypophysis divides transversely. The cell next to the embryo completes the periblem, and the other cell undergoes 2 longitudinal divisions at right angles to each other, forming a plate of 4 cells. Each of these 4 cells divide. The upper tier completes the dermatogen, and the lower tier forms the root cap. As the growth progresses, the free end of embryo becomes heart-shaped and lobed. Each lobe is a cotyledon primordium. The plumule and the epicotyl is produced in the depression between the 2 cotyledons. The plumule in dicots is terminal. The cells of the lower end of the embryo forms hypocotyl carrying radicle at its tip. Stages in the development of a dicot embryo. The suspensor cell divides transversely and produces 6 to 10-celled filament, the suspensor. The suspensor pushes the embryo into the endosperm. The uppermost or distal cell of the suspensor is large and act as haustorium, while the lowermost cell of the suspensor next to embryonal cell is known as hypophysis, which by further divisions give rise to the radical, embryonic root. Development of monocot embryo, Sagittaria sagittarifolia. The zygote divides to form a proembryo, which is a filament consisting of 3 cells, a large basal cell, a middle cell, and a terminal cell. The basal cell is present towards micropylar end. It enlarges in size, forming most of the suspensor. The middle cell divides repeatedly by a series of transverse and vertical divisions to produce several tiers of cells, which produce in succession, next to the basal cell, a few suspensor cells, radicle, rig tip, hypocotyl, and plumule, stem tip. The terminal cell divides repeatedly to produce a single cotyledon, which is terminal in monocots. There is a good deal of variations in development of the embryo in monocots. The development described above represent development in one of the most primitive monocot family, Alismaceae. Stages in the development of a monocot embryo endosperm. The endosperm is the main source of food for the developing embryo and germinating seed. It is haploid in gymnosperms and is a continuation of the female gametophyte. In angiosperms, it is formed as result of fusion of 2 polar nuclei and one of the male gametes, forming primary endosperm nucleus. Generally, the endosperm is triploid, but haploid, tetraploid, and polyploid endosperms are also found. In majority of cases, the polar nuclei fuse to form secondary nucleus before the union of the male gamete. But in certain cases, e.g., in Nicotiana, Linum, the polar nuclei remain distinct till the arrival of male gamete, which unites with one of the polar nuclei. And don't forget to subscribe for more easy biology lectures. Giz answer the following questions. 1. What is endosperm? 2. What is chalaza? 3. What is embryo? If you have any question, then ask in comment section. Thanks for watching and see you in the next class.